Pusat Riset Mikrobiologi Terapan BRIN dibentuk untuk menjalankan fungsi melakukan penelitian, pengembangan, pengkajian, penerapan, invensi dan inovasi di bidang mikrobiologi terapan. Jadi kami memiliki tugas untuk menjawab tantangan dan juga permasalahan yang ada di Indonesia maupun di global melalui pemanfaatan mikroorganisme yang ada di Indonesia. Saatnya kita berdiri sejajar dengan negara lain, karena kita memang bisa dan kita harus bisa. Dengan visi yang jelas, kuat, kolaborasi, PNMT bisa.
Sekarang yang terjadi, terobosan itu menjadi mungkin oleh yang namanya, saya ulang, oleh yang namanya ilmu pengetahuan, oleh yang namanya teknologi, oleh yang namanya inovasi.
In 2020, Indonesia entered a new national development era. On this term, President Joko Widodo administration turned its focus on developing Indonesia's human capital and mastery of science and technology through Indonesia's national research priorities. The quality of a country's research and innovation is a national asset. As a key step, the government issued Law No. 11/2019 on the National System of Science and Technology, or the Sisnas Iptek Law. By issuing the law, the government of Indonesia was signaling more attention to and commitment for the growth of the research and innovation during the next phase of Indonesia's development. 9 research fields are listed as the focus fields of Indonesia's 2017-2045 National Research Master Plan. Food, energy, health. Transportation. Engineered products. Defense and security. Maritime. Social, humanities, and other research fields. These are expected to be able to produce innovative products. In order to address institutional problems of fragmentation among government institutions and the inefficiency of the budget allocation, the National Research and Innovation Agency, or BRIN, was established in 2019 with the Presidential Regulation No. 74 of 2019 on the National Research and Innovation Agency. The President has tasked BRIN to ensure that research and development activities in the country could attract more investments to its science and technology sectors, integrating research and innovation resources, creating and open a collaborative global standard research ecosystem, and creating the foundation of a strong and sustained research and innovation-based economy are three directions of BRIN. The contribution of research and innovation to produce a variety of solutions can resolve the challenges faced by the government, industry, and society.
Pusat Riset Mikrobiologi Terapan BRIN dibentuk untuk menjalankan fungsi melakukan penelitian, pengembangan, pengajian, penerapan, invensi dan inovasi di bidang mikrobiologi terapan. Jadi kami memiliki tugas untuk menjawab tantangan dan juga permasalahan yang ada di Indonesia maupun di global melalui pemanfaatan mikroba atau mikroorganisme yang ada di Indonesia. sejajar dengan negara lain. Karena kita memang bisa dan kita harus bisa. Dengan visi yang jelas, kuat, kolaborasi, PNMT bisa.
Okay, good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Welcome to webinar series on Structural Biology 7 with topic From Sample to Structure, Insight into Phage Purification, Cryo-EM, Data Collection, 3D Reconstruction, and beyond. Held by Research Center for Applied Microbiology, National Research and Innovation Agency BRIN, today, Wednesday, February 12, 2025. In this occasion, we are honored to welcome Head of Research Center for Applied Microbiology BRIN, Dr. Ahmad Fatoni, our speaker, Rosheny Kumaran, Ph.D. candidate, University of Otago, New Zealand, and our moderator, Dr. Dede Heri Yanto, researcher of Research Center for Applied Microbiology BRIN. Ladies and gentlemen, next agenda is singing the national anthem Indonesia Raya. Ladies and gentlemen, next is opening remark from Head of Research Center for Applied Microbiology BRIN to Dr. Ahmad Fatoni. Time is yours. Okay, Pak Tony.
Okay, so I need to check. Our speaker is not here yet. Yeah, Mbak Uci. Yeah.
Oh, okay.
So we need to double check first. Okay, Narcis. Yeah. Okay, just join us. Hello. Good morning, Rosheny.
Good morning.
Yeah. Okay. I think you got wrong link.
Yeah.
Zoom link. Sorry about that.
Yes, I just came into the link. I think the previous one I had was the wrong one, maybe.
Okay. All right. Everyone is here today. Yeah.
Okay.
Thank you. Very good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. Good morning again, everyone. I would like to thank to our speaker today, Ms. Rosheny Kumaran. She's a Ph.D. final Ph.D. Student of Otago University at Ph.D. Laboratory, and I think we all here wish you very luck this year's study Ph.D. finish, and then hopefully you can join with us here in BRIN. And also everyone who has joined our webinar this morning, thank you very much for joining us. And today's topic, we are going to hear together from Ms. Rosheny Kumaran about insight into phage purification, Cryo-EM, data collection, 3D reconstruction, and beyond. We can see that I think she will be the expert, yeah, in the future, near future, yeah, specifically in phage structural using Cryo-EM, yeah, because I think in her laboratory is specific bacteriophage research, and also she, although she's in final year, but she has a lot of experience in data processing using Cryo-EM. Although in the university do not have a Cryo-EM machine, but she has worked a lot using data Cryo-EM data processing. And we will be lucky. She visited here BRIN actually last December and January, and we're lucky to have her visiting BRIN twice in December 27 and January 15, yeah, 15, and then also so actually she's already familiar with BRIN and Cibinong, yeah. Yeah, so everyone, let's hear from her study, her experience on phage characterization, and hopefully, if we are talking about bacteriophage specifically, in Indonesia, we have several researchers, but not big group, still small group, and we are going to build up to the bigger group. And this year, hopefully, we would like to make or prepare a joint proposal for research collaboration on bacteriophage specifically. And why bacteriophage is important, because it has very promising potential. We can apply this kind of creature, the small virus who infect bacteria for various purposes, I think. And this is actually our main interest, Ms. Rosheny, how we can later on utilize bacteriophage for various purposes, yeah, for medical, for environment, for agriculture, food, and many applications, yeah, in biotechnology. Yeah, that's why before we can utilize them wisely and also precisely, we have to understand, we have to learn, we have to really understand their structure, their mechanism, how these things works, yeah, regarding their function, yeah, for health, for food, and for everything, for environment as well. And yeah, I think later on Ms. Rosheny will explain why this is important regarding their specificity, and then other things, and then way forward, yeah, after we characterize their structure, understanding their mechanism, what is the next, yeah, because we cannot stop until that. We need to continue, yeah, develop bacteriophage based product for many purposes. Yeah, everyone, so please enjoy this webinar today, and hopefully if she's available, we would like to invite Ms. Rosheny again to BRIN and then work together with our researcher specific in bacteriophages, because with her supervisor, Dr. Postina, we have I think had a meeting twice or once, last year, and we agreed to have collaboration. Her laboratory will send sample to BRIN, and then we are going to characterizing our Cryo-EM, and then later on proceed the data together. And then that's how we set up this collaboration with Ms. Rosen in the future. And once she finished her PhD, hopefully, she would like to come to BRIN, just for like a 1-year project with our researcher, because as I mentioned, we have a structural biology platform for the next 3 years. So you save and stay here in BRIN.
Yep.
Don't worry about the job. And after you finish your PhD, please welcome to BRIN anytime you want. Okay. Thank you, everyone. Enjoy the webinar. Okay, I give back the screen to Mbak Uci. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Fatoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat box. Now we are going to start our presentation and discussion session, which will be led by Dr. Dede Heri Yulianto. To Dr. Dede, time is yours.
Okay, thank you very much, Chi, and ladies and gentlemen. Today we have a webinar with the title of will be delivered by Rosheny. Hello, Rosheny. Thank you very much again for coming to our webinar here, and it is a very good chance for us to learn your experience about the bacteriophage. So, Mbak Reni, could you please to show us Dr. Rosheny CV? And here, Dr. Rosheny is a Ph.D. candidate from the University of Otago, New Zealand, and she has a Bachelor of Science in first class honor from Monash University, and now she has actively write a publication with the college, and as you maybe see in this slideshow, that she published in one paper in 2024 in Indonesia. It's very good publication, I think, and she will share to us about how we prepare the sample to structure insight into phage purification, Cryo-EM, data collection, to the reconstruction and beyond. We will have your presentation maybe until 10 or 9:30. No, 10:30. Then 10, and you can continue. We will continue with the discussion. Okay, Dr. Rosheny, you are ready? You ready for the slide, please?
Yes. Yeah.
Okay, please.
Yep. So thank you to Fatoni and Dr. Dede for the introduction. As they've mentioned, I've worked a lot with phage purification and 3D reconstruction. not much in the Cryo-EM and data collection, but I'll be sharing a bit of what I've learned. So that's my title for today. So I'm from the Bostina lab at the University of Otago. My PhD is on my PhD is on the structural intricacies of icosahydroviruses. And I do this by using single particle analysis in Cryo-EM. So part of my PhD is focused on bacteriophages. And I currently have an ongoing project on a phage, but I will not be mentioning the name because it's unpublished. So I'll refer to it as phage X. So we all know that there are lots of microscopy techniques. And Cryo-ET is a technique that we use to study how proteins can interact in a cell or how cell structures are. But we use Cryo-EM, single particle analysis, to solve the structure of bacteriophages. And they range between 20 nanometers to 145 nanometers. So phages are viruses that infect bacterial cells, and they are very, very abundant in the biosphere. And we're interested in this specific class of phages, and they are called the tailed phages. So we have three different types. We have the cephalophage, myophage, and photophage. So the cephalophages here have a long, non-contractile tail. The myophages have a long contractile tail, and the protophages have a short contractile tail. So the tail here is important because it functions to eject the genome into the host. So our lab focuses on several different projects, but today I'll be talking about the part where we are interested in structural characterization of bacteriophages. And we think this is important because if we solve the structure of a phage, it will give us information on how we can develop it for potential phage applications. So there are lots of papers on how phages can be used for drug delivery and vaccines. So this would be the ultimate goal, and that's for future work by other labs. So these are the two papers that were shared before this. We work on a few different phages, but these are the two papers that we have out now currently. So the first paper is on phage N4. It is work done by Clem, our master's student, and Dr. Alice, our postdoc. And my role was to prepare and purify the phage for Cryo-EM. The second bacteriophage paper is on phage M1. This is work done by James, our completed PhD candidate, and Alice again, our postdoc. And for this paper specifically, members of the Bostina lab contributed to modeling of the protein chains because there were lots of them. And in today's talk, I'll be sharing a bit about how we prepare our phage and some challenges that we face, how we do our negative staining, Cryo-EM, data collection, reconstruction, and a little bit of the modeling. So there are several steps involved in preparing our phage samples for Cryo-EM. But the first thing is we have to acquire our phages from our collaborators, and then we can produce, purify, concentrate, and check them by negative staining. So it is common for us to grow our bacteriophages in liquid culture. And to harvest it, we would first pellet our bacterial cells by high-speed centrifugation. And then we have to filter our cell lysate with a 0.22 meter filter. And filtering here is really important because it gets rid of all the remaining cell debris. And it also ensures that we have a much more cleaner sample to work with because we don't want a dirty sample for Cryo-EM. But we did find that this liquid culture technique didn't work as well for the phage that I'm working with. So we decided to use the solid culture method. And in this method, we pour a mixture of soft agar, host bacteria, and the phage on an already existing layer. And after incubation, we would see lots of plaques. So we would want to incubate this plaqued agar with some buffer or water so that the phages diffuse into the media and we can continue with our regular purification step. So after the filtering is done, we now have our phages in this filtered lysate. And we want to be able to concentrate it so that it is cleaner afterwards. And we use the sucrose cushion method, which uses ultracentrifugation. So at the end of our sucrose cushion step, sometimes we don't really see a pellet, but it's actually there. So it'll be a very thin film. And we then resuspend this phage pellet with either buffer or water. And we did see that sometimes our phages can aggregate in sterile water. And we can prevent this by using our phage buffer that contains a little bit of gelatin. And we believe that this gelatin prevents any aggregation. So it's important to see what works best for your phage. It could be water, it could be buffer. And at this step, we would usually want a viral titer that is greater than 1 billion. And to assess the quality of the sample at this point, we would have to do negative staining. And if it is not clean enough, we could do a cesium chloride gradient purification. But we were lucky that our samples were clean and we had lots of phage particles. So we just moved on to a concentrating step where we put our phages into this 100 kDa concentrator and we spin it for a bit. And we have a very nice, we have a small volume of very concentrated sample. So we also perform negative staining on this sample. So here are some examples of some negative staining micrographs. This is the result from our sucrose cushion. And we see that there are lots of phage particles here. And after concentration, we see more phages per micrograph. And these two were at the same magnification. And we really want a lot of particles from our negative staining because during cryo-EM, we usually expect about a 10-fold reduction in particles. So this is ideal micrograph. And we can even see the characteristics of these phages. So the capsid diameter is 60 nanometers, and the tail is about 100 nanometer length. And we did this on the Joe Flash TM with a 1% PTA and neutral pH. So here's just like a brief overview of how we do our negative staining. So we add some sample onto our glow discharge grids, blot it after 1 minute and let it dry, and then add some stain, blot it immediately. And once our grid is dried, we can image it on our TEM. So now, since this phage sample is ready, we can prepare to send it to our collaborators for cryo-EM data collection. So here are our current collaborators. And we usually send them several aliquots of phage and some phage buffer, just in case, in a microfuge tube, and we parafilm it and transport it at room temperature. So we send our collaborators our samples, they collect data, and they send us our data by mail. But I'll just give a brief overview on what cryo-EM, like how the workflow usually is. So cryo-EM is a technique where we flash freeze our samples so that we can image it at a native state. Yep. And the first few steps involves putting our samples on the grid. So we apply our samples on the glow discharge grid, blot off any excess samples, and plunge freeze it into liquid ethane. So all of these steps happen at the VitroBot. And after the grid is frozen, we secure our grid into the AutoGrid ring with the C-clip. So this auto grid is then placed into the storage box and transferred into a cassette. So this is how the cassette looks like, and we can see that this is our auto grid. And this is a zoomed out version of this system. And here you have the side view. We can clamp our nanocap here, which allows us to then transfer this cassette into the nanocap. And then we take the nanocap and dock it into the Cryo-EM. So all these steps should be done in liquid nitrogen to ensure that the grid stays frozen. I also want to mention that these aren't images that we took by ourselves. And I have the sources at the bottom right. And this, for data collection, this is our grid. And this is how it looks like under the Cryo-EM. So we do have, usually with the microscope, they would have a smart EPU software, and it will usually color the grids based on categories, but it doesn't distinguish or say that one grid is better than the other. And if we zoom into a grid, we see multiple holes. Our sample would sit in a hole, and that is what we want to image. So this is a zoomed in version of a hole, and you can collect multiple images per hole. And that's just a general overview, and the software allows you to set up unattended data collection. So once data has been collected, we want to do a single particle analysis. And single particle analysis involves capturing the 2D projections of our viruses at different orientations. And then we align and combine these 2D projections to create a 3D reconstruction. So the software that we could use are CryoSpark and Reliant. Now I'll share a bit about the N4 capsid reconstruction, which is one of the phages that our lab works with. So this is work done by Alice and Clem. So after we have our data collection, we import it into CryoSpark and run a few pre-processing jobs. And after our micrographs have been processed, we run a blob pick job that picks our particles. So this phage is about 60 nanometer in size. So we tell the blob pick in CryoSpark to pick everything that is about this size. And I believe we can give it a range of size to pick from. And once the blob pick job is done, we extract our particles and run a 2D classification job. The 2D classification gets rid of junks and noise from the auto picking, because you can imagine it picks everything that looks like a capsid that is 60 nanometers in size. So this 2D classification job is very important. And we run a few jobs, and we have a few good 2D classes. And we use these 2D classes to generate our reconstructions. And our 3D reconstruction usually consists of several different jobs. And this is just a general overview of the steps that a person would do. But sometimes it can take lots and lots of jobs to actually get a good reconstruction, because there are so many different parameters that you can adjust at the 3D reconstruction step. And as I said, there are three different types of phages, and some of them have a really long tail. So the blob pick job here may not be the ideal choice to pick something as long as the bacterial phage tail. So the job that we can run in CryoSpark is the filament tracer. So what it does is that it captures the entire tail in segments. And this is how it looks like. The red dots here are the picks. And we go through the regular extraction to the classification and 3D reconstruction step. And after 3D reconstruction, there is a helical refinement job that we will have to run. And in this refinement job, it will require you to input the helical twist and the helical rise. So the twist is the rotation angle between two different asymmetric units of the tail. And the helical rise is the distance between two asymmetric units. This, the way I measure this is directly from the map in Chimera X. And this is not exactly an easy example to use, but I can use my structure because it's unpublished. So I'll just work with this. So this here is the helical rise. It is the distance between the top and the bottom asymmetric unit. And the helical twist would be this to this. So it's the angle of this region. So from the measurements in Chimera X, we can input it into CryoSpark. And for example, if our helical twist that we measured is 40 degrees, CryoSpark will allow us to give us a range that we think it is. So we can say maybe 39 to 41, and CryoSpark would run the helical refinement job, and it will tell you that actually it's 41 degree twist. So after reconstruction, we have our map. And the next step from this would be to model our protein chain into the map. So for phage M1, so we are trying to solve the capsid of phage M1. So we take the major capsid protein sequence and put it through AlphaFold. So AlphaFold here predicts the structure based on the amino acid sequence. It does this by aligning sequences with previously solved structure. So that's how we get our model. And we now have to refine this model from AlphaFold into our map. And some of the softwares that we use are Isolde in Chimera X, Phoenix, and Coot. We use Isolde to fit our main chain, side chain, fix the porotomers, fix the Ramachandran outliers. And then we put it into Phoenix that does a automatic refinement for us. And what we have to give is the map in our model. And finally, we do final refinements in Coot, where we fix any remaining porotomers, fix Ramachandran outliers. And yeah, we could just work with these three softwares. And some people have a preference of like Coot over Isolde. So yeah, just work with whatever works for you. And these softwares can be downloaded directly into your Mac or whatever laptop that we use. But we prefer to access them through the SV Grid. And they usually have lots of different softwares. I think they may have like at least 50 or even more than that. So yep, that's how we access our softwares and model our proteins. And here's just a short video on how we model our protein chains into our density map with Isolde in Chimera X. So what you see is I'm just tugging the protein chain and the side chains into density. The main chain fits really well, and we're just tugging the remaining side chains. Yep, so that is it. It's just a very short example, but it usually requires more work because you're going through a whole protein chain, and this video is just like a region. Right. So I've shared a very general workflow of how we go from purifying our sample, Cryo-EM, data collection, and very briefly on modeling. So now I'll just share a little about the two phage papers from our lab. So one of the phage that we work with is phage N4. So there has been a paper in 2008 where this group solved the entire structure of phage N4. So they have the whole thing solved. This is work done by Clem, and Clem, our master student, and Alice, a postdoc. And it was like we had contribution from Alex, a PhD student, and myself. And Alice and Clem solved the structure of phage N4 to a resolution of 2.45. Now, this is a very good resolution because the past paper on N4 solved it to 14 Armstrong. And they found that phage N4 is made up of 9 major capsid proteins and 3 decoration proteins. So it is a T9 phage. These yellow ones, blue and green, are the major capsid proteins, and purple are the decoration proteins. So Clem and Alice explored the interaction between the MCP and the major capsid protein and the decorations. And they found that the decoration proteins are utilized by the virus to increase capsid stability. So I guess a future application would be maybe like future work could be done on trying to use N4 so that it can so that it has some kind of like antigenic sequence on the major capsid protein so that it can be used as a vaccine, or it could even be used as drug delivery, because I guess that's how phage applications tend to go around. But that's something for someone else to see. And the second paper is on phage M1. So this phage is work done by Alice, a postdoc, and James, a completed PhD candidate. So this structure was solved to 3.04 Armstrong, and the whole phage structure was solved. So the capsid of M1 is made of 7 major capsid proteins. And those are the blue ones here. And it has 2 decoration proteins, which are the alpha PAW in blue and alpha claw in pink. And we see that there's more alpha claw units per asymmetric unit than alpha PAW. And they didn't see density for a large region of the alpha PAW. But when we compare the resolved regions of the alpha claw and alpha PAW, we see that they overlay really well, but they have a sequence similarity of 34.55%. So that's just a very brief overview on the capsid of phage M1 done by Alice and James. But the focus of this paper is on the ejectosome. So Alice and James here put in a lot of work to reconstruct all of these regions. And here's a diagram of the workflow that they used. But I won't be going through it because they've used a lot of different techniques. And I think if people want to learn more, they can always go read this paper. And this figures in the supplementary, so it's accessible. And if anyone has any questions on phage N4 or M1, you can feel free to just reach out to the first authors because they know it really well. And yeah, I guess that's all that I have to share. And I just want to say that I've presented a lot of work done by Alice, Clem, James, and some for myself. And most of the projects here had lots of contributions from Francesca, Georgia, Nick, and Alex. I also acknowledge our collaborators for all the Cryo-EM and data collection work that they do for us. And Thanks to my supervisor and co-supervisor, Mignia and Laura. And here are my references if anyone has any papers or things that they want to read based off my presentation. And I'm happy to take questions. Thank you.
Okay, thank you very much, Rosheny, for a very nice presentation, and I hope everyone can got a good experience from Rosheny how to prepare your bacteriophage samples and how you analyze the structure. Yeah. So we now open the question from the audience. Please, if you have any question or comment, you can raise your hand and open mic, or you can write the question or comment in the chat. Yeah. Anyone have a comment or question?
I'm open to questions or comments. So,
Dede, may I have question?
yeah.
Hello, can you hear
Hello?
my voice?
Yes, but it's very, the volume is really low, but I can try.
Yeah, please.
Okay. I have one question related to the preparation. I'm Agustin from Brawijaya University, Pak Dede.
Oh, Bu Agustin, yeah.
Yeah. Okay. Thank you for the presentation. This is a very interesting presentation. And I have one question related to the how to prepare the bacteriophages for the cryo-EM in terms of the concentration. So how much concentration of the phages for the cryo-EM, for the accurate cryo-EM? Because in my case, I'm very difficult to get the high amount of concentration of our phages. Okay.
Right.
This is my question. Thank
Okay. Thank you so much for the question. I'm not too sure how checking concentration of bacteriophage works because the bacteriophage itself is made up of many different proteins, and like, for example, one of the phages I work with has, I think, 13 different proteins. So I don't think the protein concentration reflects how many phages we might have. So what we do is we do a plaque assay to determine the viral titer. And we've seen that a viral titer of more than 1 billion would usually give us enough bacteriophages on the TEM and for cryo. So yeah, it will be the viral titer that would be very helpful to know. Yeah, I hope that answers your question.
Okay, thank you.
Yeah, yeah.
Okay.
Any question, Bu Agustin?
I think it's enough.
Okay.
Okay.
Yeah.
I'm happy to answer any other questions via email if anyone
Okay.
has any questions.
Yeah.
Okay. Okay, Bu Ira, yeah, Bu Ira and Bu Fitri, please open your mic. I think it's okay. You can open your mic.
And just a simple question, Prof. Dede. What is the size of the bacteriophage? How many kilodalton? So I use S.Si. Thank you.
Yeah, Rosheny, sometimes the bacteriophage is big, and how we like manage our SPA, because SPA is sometimes small or until maybe 1000 Dalton. Yeah, but how is the size of the bacteriophage, and how you manage? For example, did you do one by one part of the pages, or what will you do?
Yeah, so the bacteriophage itself is very big. In terms of the protein size, that will be a lot to calculate because the capsid is made up of so many copies of proteins. But for single particle analysis, because the phage is really big, you have the head, you have the neck, and you have the tail. So for single particle analysis, we will work on all of these parts individually. So the capsid reconstruction will go through its own pipeline, the neck reconstruction will go through its own pipeline, and the tail will have its own. So we don't do the entire bacteriophage reconstruction at once.
So part by part, you
Yeah.
yeah, okay. So yeah, I think this technique is a very we don't have any experience how do the reconstruction part by part of the page and then getting together become the big structure. I think it is very nice experience if you can teach us in the future maybe we during our collaboration.
Mhm.
Okay, thank you so much. And Bu Fitri, please, for the question.
Okay, thank you, Rosheny, for your nice talk. So I just want to, you already show us about the general workflow of the data analysis part, but actually, I wonder that you also already mentioned that when doing phage constructions, you're doing part by part. And I wonder that how many particles that is optimum to construct the phage. For example, it's like in during your reliant, for example, it's like how many particles that you use to construct your proteins. And also did you change the focus several times to obtain those particles or not? Thank you.
Thank you so much for your question. So I have tried using Reliant, but I work mostly with CryoSpark at the moment. And I don't think that particle number really influences resolution because I've had like large particle numbers, but the resolution is not that great. But for some samples, you have very low particle numbers, but the resolution will be up to like 3.5. So I don't think we can like relate particle number to the resolution that we get. And for your second question on the defocus for collecting images, I'm not too sure about that because we send our samples to the Cryo-EM facility and they are the ones who are usually responsible for like collecting our data. So yeah. Thank you.
And okay, any else, Bu Fitri?
No, I think
Okay. Okay, so Rosni, I have no experience about how to prepare pages. Yeah. So, according to your experience, is it difficult to purify the page from the host or from the microbe, or is it difficult or not? According to how difficult to prepare this? Sometimes because sometimes I receive the sample from the customer and the pages is not good or the sample
Yep.
the purification is not good. So I don't know how the difficulties to purify the pages from the sample.
Right. Okay. So like a part of my PhD required me to prepare like virus-like particles, and that purification workflow was really, really difficult. So I personally find this bacteriophage purification workflow to be much more easier compared to like the other purifications that I've done. So yeah, I would say it's much more um, simpler to do it. Yeah.
By only
It's
the filtering.
Yeah, the filtering is also really easy to do. It's just that you have to like replace the filter multiple times. So I think that would be like spending money on resources. Yeah, but it is a pretty straightforward and easy workflow to do. And you just have to make sure that we have enough viral titer. Because if we don't have enough pages, then you won't really have anything. Like you won't have much pages to collect in Cryo-EM.
Okay. Yeah. Okay. We have a question also from Pak Yoga here. And Pak Yoga, please, if you want to open mic.
Okay, thank you. Yeah,
Thank you.
could you hear me?
Yes, I can.
Yes, I can. Okay. Yeah, I didn't experience with Cryo-EM microscopy, but only TM. I want to ask about any possibilities to synchronize Cryo-EM data with biomolecular, like DNA or any RNA data for structural function of phage particle.
Right.
Any possibilities?
Um,
Thank you.
Thank you. Thank you for your question. I'm not too sure about that question, so I can't really answer that. But yeah, I could look into it and see.
Yeah, I think for yoga, yeah, we can not only synchronize, but we have to fitting our cryo-EM image data from the cryospark. The data from cryo-EM is we call the image, and when we obtain the image, maybe around 5,000 images, and then we processing the data using the Reliant or cryospark. And after we validation and refinement, then we always to fit our we call our structure here with the data from sequence, our DNA, our RNA, amino acid sequence, something, and then we fitting using the like a Phoenix or a code to get the structure is validate. Yeah, yeah. Of course, we synchronize with this data.
Oh, good.
Yeah.
Thank you so much.
For example, from the upper fault, from the upper fault we get the model structure, and then we fitting this model structure with our real structure obtained from the cryospark. Yeah, fitting together.
Wow, thank you. Any possibilities to collaborate with BRIN to cryo-imaging?
Yes, of course, with the Patoni and also with the Cryo-EM lab, yeah, we open for this collaboration.
It's nice. Thank you so much for
Yeah,
information
thank you.
and answers. Thank you.
Thank you so much, Pak Yoga. And any question from the audience?
Yeah,
Pak
Pak Dede,
Kapus?
Rosheny, yeah, just want to add about the collaboration things. Yeah, I just remember that in the last email, Dr. Posen mentioned about the MTA, material transfer agreement, because you would like to send some samples. I think 5 or 6 samples ready for Cryo-EM from Otago University, from your library actually, but I haven't heard yet, from him again regarding the MTA. I have sent the template from BRIN because actually this year BRIN just created new team specifically with the duty of discussing about material transfer agreement with our partner from overseas. Yeah, but I think we can later on. I will try to resend email to Postina and then discuss about the MDA, because hopefully you can send not only your sample, but you can come to print again and then do together with us here characterizing your sample using our Cryo-EM. Yeah, so we have Cryo-EM, but no experience yet much on bacteriophage. You have, I think, a lot of experience on bacteriophage processing data, but I think we can complement each other. And I saw here I think your colleague, I saw Alex Crowley, your colleague from Otago. Yeah, I saw. Yeah, his or her name, but yeah, thank you for joining us, and hopefully you can also come together with Rosheny later on to print it. Then regarding the collaboration, not only with Otago, Bapak Ibu, any campus university from Indonesia, Pak Yoga, just contact Pak Dede or us, to build up new collaboration, specifically utilizing our Cryo-EM. Yeah, thank you.
Thank you. Mr. Ahmad, thank you.
Okay, thank you so much. And is there any question from the audience? I think Dr. Rosheny, you also okay, right? If someone from the audience or contact you by email or some or
Yeah,
asking
yeah.
you something about the
Yeah, I'm happy to
bacteriophages.
answer
Okay.
answer any questions through email. Yep.
Yeah, thank you so much. So yeah, maybe if you don't have any question again, I think it is good for us to end of this webinar, and I will give
One
again
question, Pak
to
Dede ya.
the. Okay,
Ya
Bapak
Pak Dede.
Otoni.
Yeah, Rosheny, actually
Yep.
for the moment we have one or two project on pherophage, but I think the PI is not here. Yeah, I think he shouldn't miss this chance to discuss with you, but hopefully he will contact you or discuss with you. Yeah, maybe through email or later on when you come here. But I would just
Yeah.
mention if I think we want to accelerate research on bacteriophages as a Prof. Agustina led previously from Perwita University. This year we would like to create, we call it research collaboration center, yeah, for bacteriophages. And in that case, I think we need your expertise here. So inviting you, hopefully it is still possible, yeah. Like we later on we can run a hands-on workshop from purification step until we submit to Cryo-EM and then proceed the data after Cryo-EM. Yeah, I don't know. Maybe we can after this we can discuss in more detail, like a schedule how many days that we can do this kind of workshop, and if possible we can invite you again to BRIN to run the workshop, or we can like do online workshop first if possible. Yeah, so we after this we would like to invite also researcher from university and also from other research centers, because we recognize there are maybe around 10 researchers, but from other research centers maybe they're not joining us today, but I would like to invite them, and then maybe we can run online workshop if you don't mind, or after you obtain your Ph.D, then as soon as possible I will invite you to BRIN.
Yeah.
Yeah, I just want
Yeah.
to add that. Yeah, that matter. Thank you.
Yep. Yeah, thank you so much for sharing and having me speak here today. And I also want to mention that we were on a conference like quite recently, so that may be why M.Eng hasn't responded to the emails, but I'm sure he'll get back as soon as he can.
Sorry, one thing. Is your lab also running like a workshop, hands-on in your laboratory, like a annual workshop, something like training? Or
I don't think we have any workshops for like external, but then we do like help each other and like train each other out sometimes if required.
okay, because if you do like that, we can send up to 6 researchers of BRIN to Otago University to get the training, on the data processing, for example.
Right.
Yeah, but maybe if you can, maybe we can make like a syllabus for our training when we already signed the collaboration document later on. Yeah, I think we can set up this kind of a workshop or training in your laboratory or at Otago University. Yeah, in the future. Yeah, thank you.
Okay. Yeah, thank you. Yeah, I'm sure M.Eng would be happy to like discuss anything. So
yeah. Okay. Cool. Thank you, Rosheny.
Thank you. Yeah, thank you so much.
Yeah, thank you, Dr. Rosheny. Yeah, we hope that we can implement our collaboration between BRIN and Otago University in many fields. Yeah, I think in Phage Purification, yeah, by workshop or training together. Of course, we have BRIN has a good Cryo-EM facility, and we open for the collaboration with Otago University for sending sample and we analyze. the data acquisition, and then we together data processing between BRIN and Otago University. Okay, thank you very much again, Rosheny, for
Thank
very
you.
nice sharing about your experience in the page purification and until processing that data processing. Do you have any something to say before finish?
No, I don't have anything to say. I just want to thank Pak Tony and Dr. Dede for the opportunity for me to like share what our lab does.
Okay.
Because yeah.
Okay, thank you.
Yeah.
So
Heri.
yeah.
Thank you so much.
Please, Mbak, itu ya. Thank you.
Okay,
Mbak
thank
Uci
you.
ya, please.
Thank you, Dr. Dede, for leading a very interactive discussion, and thank you, Ms. Rosheny Kumaran, for a very interesting presentation. And as a token of our appreciation, we will give a certificate for Mr. Rosheny Kumaran as a speaker. Thank you very much for sharing your valuable
All right, great.
knowledge.
Thank you, Ms. Rosheny.
Thank you so much.
Okay, ladies and gentlemen, we have finally come to the end part of this webinar. Before I close this session, I would like to ask Head of Research Center for Applied Microbiology, BRIN, to give us some short closing remarks to Dr. Ahmad Fatoni. Thank you.
Okay. Again, thank you very much for Ms. Rosheny for your sharing knowledge and experience on bacteriophage research. And this is just the beginning, and hopefully I can see the long and future collaboration with your team on bacteriophage research and utilizing our Cryo-EM facility. And thank you very much for everyone who is joining us today. And so please feel free to contact us, Pak Dede, or even email or contact Ms. Rosheny regarding the bacteriophage research. Yeah, thank you, and close this webinar. Assalamualaikum warahmatullahi wabarakatuh. So I give the back to Mbak Uci.
Thank you, Dr. Ahmad Fatoni. Ladies and gentlemen, this is the end of our agenda for today. I'm Andalusi Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. And for your information, you can check out our latest information about PRMT at our social media on Facebook, Pusat Riset Mikrobiologi Terapan, and Instagram at PRMT_BRIN. You can check the CV or our speaker and the contact on the highlight story on the Instagram, and you can also check the recorded webinar on YouTube at PR Mikrobiologi Terapan BRIN channel. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, everyone. Bye-bye.
Thank you, everyone.
Thank you, Ms.
Thank you,
Rosheny.
Master.
Yeah, see you in
Thank you.
Cibinong, yeah. I know this year,
see
the
you.
end of this
Thank you,
year,
Pak
next
Ahmad,
year.
Pak Dede.
Yeah, best of luck
Yeah.
your study. All right.
Yeah. Thank you so much.
Thank you. Thank you. Bye-bye, everyone.